The PIDGIN Project is an interdisciplinary initiative at Hasselt University, and this project brings together experts from different fields such as oncologists, pathologists, biomedical sciences, but also data scientists. Our ultimate goal is to find a more precise way to diagnose lung cancer based on tumor heterogeneity. In this project, microscopic slides are digitized using a slide scanner. And the main advantage of using these digital images is that they can be accessed from anywhere and also processed using a computer. And this allows us to use these images to generate AI algorithms that can assist in making an accurate diagnosis. And the main advantage of these AI tools is that they can detect abnormalities in the tissue that cannot be seen with the human eye.
A pathologist looks at the tissue slides that we are recording in a clinical way. So it means that it rates the overall image of the tumor and decides what type of tumor it is. What we will do is we use computers and AI technology to look at these slides in a subclinical way. So that means that we will look at the level of pixels and see if there are differences between healthy cells and tumor cells. Machine learning is a very important aspect in this project, and it will be used in several ways. First of all, we have an algorithm, it's a UNet, that we will use to detect the cells in our high-resolution image. Doing so, we have the entire cellular makeup of the tumor, but we still don't know what type of cell we're dealing with. That's where the second use of machine learning comes in, because in this project we will have not only the high-resolution tissue slide, but we'll also use sophisticated colorings of our tissue, which allows us to have a ground truth value of what cell type it is. For example, we have a coloring so to understand which type of whether a cell is a blood vessel or not. So if we now feed this information to an artificial neural network, we can train the network so that it can recognize, based on the high-resolution image, which cell type they're actually looking at, without the need of this sophisticated and expensive coloring.
So the PIDGIN project provides high-resolution images from which we can extract the location of each cell as well as features of those cells and its neighborhood. With this information, we develop statistical models to quantify tumor heterogeneity. This means that we investigate the spatial interaction of the different cells. Understanding how cells are distributed within the tumor then helps in identify patterns related to disease progression or treatment response. Digitized slides enable an objective and reproducible quantification of tumor heterogeneity. This is an advantage because then we can automate the analysis of cell composition and the spatial relationships, and these insights can then help the pathologists to more accurately interpret the slides.
The VSC provided 3 key enabling services for the PIDGIN project. First of all, we have the Tier 1 data that allowed us to store the data at the infrastructure of the VSC. Second, we have the Tier 1 compute that allowed us to train the deep neural networks and come up with a solution of classifying our high-volume images. 3, to train these networks, we need to have a lot of RAM memory, and we also had that at the VSC infrastructure.
VSC gave us a great solution to store and share our data, but also at the same time do complex computations on this data.